have people log in. So we're just going to take a few minutes this morning to do a little housekeeping, reminders of a poll, and we'll do a quick networking session and then jump into our presentations. Got a great lineup for you today and for the rest of the week. So just a little intro. Since 2001, RiversEdge West has been hosting this annual conference that focuses on impacts to riparian habitat in Southwest US. And one of the main things that we like to do at the conference is, bring people together to discover techniques, scientific findings, lessons learned that will hopefully make our research and our restoration more successful and long-lasting. So this week we'll have a great lineup of live pre-recorded sessions. The presenters will be with us to do Q&As. We'll have some field tours, we have some demonstrations and workshops, and we'll try to do some virtual networking as well. So that's kind of what's in store for this week. Some of the sessions are going to be talking about changing conditions, projects and lessons learned, tools, technology, and monitoring, and of course, research. So that's what we have in store. So first off, I want to thank our sponsors. We could not do this without our generous sponsors. So I would like to just take a moment and thank all of the foundations, consultants, local, state, and federal governments, corporations, and NGOs that sponsored this conference to really make it happen. So thank you so much for your commitment and your support of this event. Moving on, today's agenda. Well, before I get to today's agenda, let's have a little fun. We're going to launch a poll just to get everybody started. So I'm going to launch a poll to everyone. So if you had to choose, what would be your spirit animal? So if you could click on what your spirit animal would be, we will we'll share the results. Okay, we got a few coming in here. We're at about 40% response rate. It's a race between river otter and raven so far. Okay, looking good. Some people are earthworms, octopus, no elephants. Seriously, no elephants? Don't they have the best memory? All right. We're almost there at about 90%. Okay. It looks like river otter won. So that's how people chimed in. River otter and raven. And then, split between dog, cat, bear, earthworm, and octopus. So, okay. We're going to do one more. This is a little bit more serious. Let's do a quick poll to see who attended last week and who attended, both days, or who attended the first time this week. So let us know where you're at. All right. Okay, going to give it another second here. And okay, I'll share those results. So it looks like 65% of you attended both days last week, and about 20%, is this the first day attending? And then 15% attended one day of last week. So, well, thanks for sharing that information. That's helpful to know what your spirit animal is, and if you joined us last week or new to the conference this week. So, okay, moving on. Like I said, today's agenda, we're going to do a quick breakout group this morning. We're going to talk about changing conditions. We're going to have a break for lunch, about an hour. Just a reminder that we do have breakout rooms available if you want to connect with your colleagues, or if you need a room to just have a meeting with somebody that you've been meaning to connect to. Reach out to us and let us know if you need a specific room, and we'll set you up with that. Remember that you can chime in and and let us know in the chat box if you do need, one, a breakout room, or two, if you're experiencing any issues. We have lots of people behind the scenes helping out with IT and support. So just let us know if you need any help. Just enter your questions or issues into the chat box, and we'll have somebody jump on that for you. So this afternoon, we're going to talk about project examples and lessons learned, and then just a final discussion, wrap up, and networking. You can find our full agenda for not only today, but the rest of the week at the link on the screen. We'll enter that into the chat box, so you guys can have that link and access the full agenda. So one quick housekeeping thing, we're all accustomed to Zoom. The ever important mute button is we just want to make sure that everybody stays muted while the presenters are happening, so we can have their undivided attention. We'd love to see faces, so if you want to keep your video on, that would be great, but we understand that it tolls on the bandwidth. So if you are having trouble with the internet, feel free to click off your video, but just a reminder to keep your Zoom muted. As a reminder, we have continuing education credits for some organizations and parts of our conference, from Society of Ecological Restoration, the Wildlife Society, Society of American Foresters, and Society of Range Management. If you are seeking continuing education credits, please reach out to John Leary. His email is here on the screen, and he can get you connected with getting those continuing education credits accounted for. And want to put a plug in for our evaluation. This evaluation really helps us improve on the content, the links, the format of our conference. So please fill out this evaluation. Be candid with us. Let us know what you like and what you don't like, because we want to improve this conference to benefit you. So we'll go ahead and put that link in the chat box as well, so you can directly connect on that. Even if it's for one day, let us know how we're doing. But if you're attending multiple days, make sure you get us that evaluation by the end of the conference. Okay, well, with that, I think we're one last thing I wanted to mention before we do a network question. We are recording the sessions, and we are posting those onto a Vimeo link. You all will receive a code to access all those presentations. Just be patient with us. It takes some time to get the editing done and those linked up to the Vimeo. But we know that many of you have already accessed those videos, but we will have those up as soon as we can. So we are recording all the sessions, and they will be posted for playback. So with that, I think we're going to go ahead and launch into a bit of a networking. Let's get to know each other a little bit. As last week, we're going to break out into, maybe about 3 or 4 people per room. Introduce yourself, who you are, where you work, and then I guess just connect with people and what were you hoping to get out of this conference? Maybe those people in the room can help you with that. But if not, let us know. We'd be interested in hearing what you are hoping to get out of this conference. So we'll go ahead and break out into those networking rooms, and we'll see you back here in just a few minutes. Okay. Well, hopefully we solved the world's problems in that short 10 minutes. if nothing else, I hope we met some good people and connected with some of the other participants for our conference. So I think with that, we will go ahead and get started. There we go. I didn't even follow my own rule. I'm sitting there chatting and I'm on mute. So, well, I hope everybody got to meet some of the other participants for the conference and solve the world's problems in that short 10 minutes. So we're going to go ahead and get started with our first block of presentations. First block is changing conditions. I'd like to welcome our first presenter, Adrian Oglesby, with the University of Mexico Transboundary Resources Center, and Paul Tichon with Audubon, New Mexico, and they're going to talk about the changing conditions on the middle Rio Grande.
Sorry, technical difficulties. Let me see what I can do here. I got it.
Bear with us while we work on the audio and video.
Hey, good morning, everyone. Hope it's a beautiful Tuesday morning wherever you are out there on your favorite river. My name's Adrian Oglesby. Want to thank Rusty and Kara and the rest of the RiversEdge West crew for having me here at the conference with you all today. I do want to start off by saying that I am giving a different presentation than that which was advertised. The presentation that Paul and I were going to give this morning was essentially given in late January at the New Mexico Water Dialogue Conference. So if you want to learn more about that topic, feel free to visit the Water Dialogue's webpage where you can view that. But we thought we'd give you a little bit of variety and talk about changing conditions in the middle Rio Grande Bosque. So there's my contact information. I work at the UNM School of Law at the Transboundary Resources Center, where we primarily focus on water and natural resource issues in New Mexico and the Southwest. And to give you a sense of place about the middle Rio Grande, first I should say that I will be referring to our riparian forest as the Bosque. That's how we do it here in New Mexico, so bear with us. The middle Rio Grande is an incredibly complicated place to work. We've got a lot of irrigated acreage. We've got numerous Native American pueblos. We've got 3 national wildlife refuges. They have trees in them as well. I see my misspelling there. And a couple of cities. We've got a Supreme Court case brewing down south with Texas. We've got endangered species. And I'm going to talk about a lot of the traditional problems that you all are experiencing on your river systems as well. One thing I wanted to point out is that the current river, as we know it, has been around for about 5 million years in this valley. The Bosque itself, probably for about 2 million years. And then for those of you who weren't aware, the middle Rio Grande is a rift valley, like the Nile. We're only concerned with what's happening up in that little bit of basin fill at the top right now. This valley has been farmed for a long time. It's actually the longest continually inhabited area of the United States, particularly the floodplains. The Bosque is one of the largest contiguous cottonwood willow forests left in the American Southwest, possibly the largest. And this Bosque has been through a lot over the millennium. One example is after the Spanish came in the late 1500s, there were some incredibly cold winters, and the Bosque, for the most part, throughout the Middle Rio Grande, was completely denuded for firewood. So again, this is a system that has experienced all sorts of changes, not the least of which was massive development in the upper watershed in the mid 1800s. First white settlers came into the San Luis Valley around 1851, bringing with them lots of sheep and a lot of desire for irrigated agriculture. So essentially, we saw a collapse of the upper watershed as the river was drained for farming, and a lot more topsoil was loaded into that river because of the grass denuding that was happening due to all those sheep up north. Down in the middle Rio Grande, the valley flattens out considerably. So as that sediment came down in that weakened river system, it began to erode our river in some years, 10, 12 feet in some areas. That, of course, caused dramatic increases in flooding, river avulsion, and high groundwater. We had our own population growth happening at the same time down here in the middle Rio Grande, and folks were moving in closer to the river and constraining it. And the floods that occurred, in particular, 1884 was just a massive flood, 100,000 cubic feet per second flood that raged for 9 days. Massive destruction of land and farmland, urban area. Had other big floods. 1904 was a particularly bad year. 1941 is one of our more notorious years in flood history. And the responses, of course, are things that you're all familiar with. The building of levees, the digging of drains, the raising of dams on our tributaries, the lining of the river with jetty jacks to constrain it, lock it in place, and the imposition of the main stem dam in the 70s, which I'll highlight here in a moment. Here's a shot from the West Mesa to give you an idea of how dynamic this river system was before we really tried seriously to harness it. I'd like to show you this shot. Not a terribly great aerial, but it is purported to be one of the very first aerial photographs taken in the American Southwest. So a little historic value there. Here's another shot of that same area. You can see the urban encroachment. This isn't much later, about 10 years later. But you can see still, braided channel, nice wide floodplain. This is Cochiti Dam. This is one of the largest earthen dams in the world, and it sits at the top of our valley now. Cochiti was put into operation in 1975, of course, to hold back those floodwaters that had been so destructive in previous years. Cochiti really is considered sort of the nail in the coffin of the natural hydrograph in the middle Rio Grande. With this main stem dam, our snowmelt pulses were scalped, and the river became much more managed in relation to its natural functions. In addition to that kind of constraints, here are the Jetty Jacks. Thanks to Paul Tashin for this photo. I see the credits a little below the screen there. Thousands of these Jetty Jacks were put into place up and down the river, locking the channel into place. And really, a major constraint for us. One of the interesting things about the Jetty Jacks being put into place is that one of the flood control acts in the mid-century mandated that the middle Rio Grande be no more than 600 feet wide by law. So that's an interesting constraint as you're trying to think about a meandering river system. Here's some comparative aerials to give you a sense of the constraints in the channel. I will point out 1957 was a particularly dry year, but you can see how the sinuosity has really been reduced. This is another image of the constraints on the channel. Again, you can just see how narrow the channel has become over the years. Here's an image that helps you understand the urban encroachment. That's the red, and it is a lot. We have really hemmed this river in with our urban development, and I stand guilty of that. I like the fact that I live a couple blocks away from the river in the ancient flood zone. Here's an aerial from 1935 of the Albuquerque South Valley there on the left. You can compare it to the image from now on the right. Again, very narrow, constrained, a much narrower riparian corridor. But you can also see a denser riparian corridor as we now deal with exotics such as saltcedar and Russian olive. Here's just another image to help you understand the river channel narrowing and perhaps a bit of the story of a loss of our wetlands. This is right at the Central Avenue Bridge in the middle of Albuquerque, 1934, I believe. And here's what it looks like today. Again, a lot less water, a lot narrower channel. Essentially since the installation of Cochise Dam, we've gone from the problem of having way too much sediment coming down the river to now not really having much at all. So we're experiencing sort of the inverse problem that I was first describing with instead of degradation of our channels, which is leading, of course, to increased disconnection from the floodplain. And then here is image showing you the wetland loss in the reach south of Albuquerque. That's 1935 to 89, give you a sense there. But I don't want to depress you too much. There is hope. We still do have floodplain connectivity. Here's a photo taken by John Fleck showing some of the flooding. I believe this was the 2017 overbank flooding that we experienced. And then we experienced more in 2019. We just had some really good water years and had some high flows. This is a shot from the 2019 overbank flood taken by Dr. Eichhorst over at the Bosque Ecosystem Monitoring Program. These intrepid citizen scientists are out looking for their precipitation gauge, which I think is funny given their situation. So lots of planning has been done in the Middle Rio Grande Bosque. We have learned a lot about this forest system over the last few decades. This publication came out in 1993. It was written by a committee of really great water managers, but led by Dr. Cliff Crawford, who we've lost now, but Dr. Crawford, it was a huge influence on many of us in this valley, and I sort of referred to as the dean of the Bosque. This document I really consider to be the Bible of Bosque Management, and it was built largely on the concepts, not explicitly at the time, but on the concept of dynamic patch mosaic, and this opened up our eyes to how ecological processes happened in this valley, the dramatic importance of floodplain connectivity, and has been very influential on our water and land managers. Also in 1993, another big shift was that management of the 30,000 acres of Bosque was largely moved from our irrigation conservancy district, the Middle Rio Grande Conservancy District, to the Rio Grande Valley State Park, which is operated by the city of Albuquerque and New Mexico State Parks. That opened the door for increased restoration opportunities and increased intensive management of the Bosque. I wanted to highlight this planning document that was written in part by Yasmin Najmi. So shout out to Yasmin, our current RiversEdge West, New Mexico board member. This really hammered home the importance of the dynamic patch mosaic. Is a management concept for the Bosque. This notion of mimicking natural and destructive forces, of the natural, destructive and creative forces that the river expresses, how we can mimic that, in our Bosque management. And these planning documents, in addition to many, many more, led to the effort in 2015 to establish a conservation action plan for the entire Bosque, for the full reach of it, not just one stretch, not just one neighborhood, but looking at this entire riparian forest as a whole, which really wasn't being done before. Different jurisdictions were doing their different projects. And so we wanted to look at the forest as an entire unit. The notion being that, for example, we were seeing a lot of cottonwood pole planting being done as a restoration technique, but not a lot of establishment of saltgrass meadows. It might be that using our analysis, some jurisdictions might decide that there are other types of habitat that they want to try to restore to complement the extensive cottonwood pole planting that's already going on. So here are conservation targets for those of you who are familiar with conservation action planning. These charts should either bring you great joy or angst. So we picked the riparian and wetland vegetation communities, native bird habitat, native fish habitat, wildlife corridors, and ditch and drain habitat as our conservation targets. Ditch and drain habitat really, is important in terms of the recreational uses by our urban constituents, the non-farmers who make up the urban part of the Middle Rio Grande, put a lot of stock in what they see along the ditches. So that is important. And then here we've listed the threats that we've identified down the left. And I just want to pause for a moment and thank the folks who were involved in the 2015 effort. It was a partnership with the Natural Heritage Program in Colorado, Lee Grinnell and Renee Rondeau. We partnered with New Mexico Natural Heritage, Elizabeth Milford, and in particular, Esty Moldavan. And Yasmin has been a big part of this process from the beginning. The 2015 effort was initially funded with some seed money from the Middle Rio Grande Conservancy District. And so in 2020, we had to pause our effort for a few years there due to funding and everyone being busy. But in 2020, we were able to scrape together a few pennies and bring our team back together here in New Mexico, Yasmin and Esty in particular. We added Jonathan Tyrell, who recently graduated from New Mexico, someone who started in the Bosque Ecosystem Monitoring Program in 4th grade, so brought a tremendous amount to this process. And we held one big meeting and then pulled together subcommittees. We've got over 50 participants, scientists, decision makers, leaders in the community. So very grateful to all the folks who have participated in this, both in the 2015 effort and now in our ongoing sort of reinvigoration of this conservation action planning effort. So far, some highlights from our 2020 work here on the screen. We've pulled together our committees around these targets that we're trying to protect. Some incredible expertise. And our hope now is that we can turn to that last very important stage of taking what we've learned, taking all the recommendations that we've developed, and communicating these effectively to our decision makers so that we can see some real implementation on the ground of the dynamic patch mosaic from the top to the bottom of our valley. So with that, I'm going to end again with a photo from Esty Moldavan. One more thanks to Esty, who really has done the majority of the heavy lifting in this project over at New Mexico Natural Heritage. So with that, I'll thank all of you for your attention, and I will stand for questions. Thanks, everybody.
Thank you, Adrian. Appreciate the presentation, and we appreciate the years of service that Adrian gave to our organization as a board member. So we do have Adrian here now. I want to remind everyone, if you do have a question for Adrian about his presentation, please enter it into the chat box right over here. You can enter the chat box and by hovering over the toolbar at the bottom of your screen. So Adrian, I'll just kick off with a question.
Sure.
You know, that that slide that you had where the the flows were downgraded to fair to poor. What is the long-term trajectory for the Bosque? Is it naturally regenerating, or is there going to need to be continued help, or what's the trajectory here with Bosques that are so important in Mexico?
Yeah, unfortunately, it is, as I said, a really heavily managed system, and we do have a lot of floodplain disconnection throughout the system. So yeah, maintaining a natural bosque is something that is going to take a lot of intervention. We have a lot of intervention happening already. Most of our agencies are working hard on preserving this great feature of ours, but yeah, it's not going to sustain itself.
Yeah, I was afraid of that.
Yeah.
As the river channel narrowed, was there a significant increase in woody vegetation, both native and non-native, throughout the years?
Well, yeah, I mean, you got to keep in mind that this, they say the Rio Grande used to be a mile wide and an inch deep.
Right.
So this river used to move around a lot, and was wiping out stands of trees and reestablishing other stands elsewhere. So yeah, as we've moved it down into this 600-foot wide channel, of course, what we've seen is the increases in density, a lack of the destructive force. So we're losing those open areas, or we're not creating those open areas anymore. And yeah, we have we work hard at fighting it, like all of you guys do. But we've seen a lot of salt cedar in this valley, and Russian olive as well, of course, which is added to that density problem. When I first started going down to the Bosque, in the mid '90s or late '90s, it was just a wall of salt cedar in a lot of places. If you wanted to get to the river, you had to crawl. And we've opened a ton of that up, which has been really great to see. So we are making progress there.
Right. Couple quick questions, and we'll move on. Is the beetle helping or hurting your mission?
I wouldn't say either at this point. I mean, we do have the beetle. Came down through the Rio Jemez into Santa Ana Pueblo, and then spread up and down the middle Rio Grande from there. I think we've seen more kills down sort of south around Truth or Consequences. But it I can't really say it's been a help or a hindrance at this point. Yeah, it's been more of a curiosity.
A lot of areas are kind of wait and see as the beetle persists on the landscape. All right, one more. What proportion of the current dense looking vegetation is native versus non-native?
I don't know. And of course, it depends on what area you're looking at. But frankly, I don't have an answer to that. I should probably remind folks that I'm the lawyer in the bunch.
Well, Adrian, thank you so much for your presentation. If we have any more questions for Adrian, hopefully he'll be around. If not, we'll try to facilitate getting answers to those questions. So Adrian, thanks so much.
Hey, no, thanks, Rusty. It's good to see you, and hopefully we'll all get together for a float later in the summer.
Absolutely, always up for that. So
right on.
Okay, we're going to keep it moving on. Next up is Alex Getz from the University of Denver. His presentation is recovering salix following the Tamarix removal.
All right, thank you for watching this. This is a presentation about salix recovery following Tamarix removal in riparian systems of the western U.S. My name is Alex Getz, and before I get started, I just want to mention that my co-author Ian Moffat will be available on the Q&A session as well to answer questions. So just to give some really brief background, I'm sure everyone is pretty much familiar with this dynamic, but the southwestern willow flycatcher is a federally listed endangered bird that uses Tamarix often as nesting habitat because its native habitat is no longer present in a lot of these areas, and defoliation by the Diarrhabda beetle therefore may reduce these flycatchers' reproductive success because it is reducing its nesting habitat. So our research questions today are, first of all, whether it's possible to increase salix cover following Tamarix removal in such a way that we don't see a net loss of flycatcher habitat. Second, whether there are particular restoration methods associated with increased salix cover following Tamarix removal. and also whether there are particular environmental variables that are associated with increased salix cover following Tamarack's removal. So our methods for this, we used a large data set from riparian restoration projects across the Southwest. We had 243 treatment sites, 93 desirable reference sites with mostly native vegetation, and 79 undesirable reference sites with mostly invasive vegetation. And in addition to plant cover data, we also had data on treatment methods and abiotic conditions at each site. And this data came from a large data set that the lab has been doing a lot of work on in past years. And so I'll talk more about this later, but we conducted separate analyses on all salix species as well as just Salix exigua. So our study area encompassed quite a bit of the Southwest. You can see it's clustered mostly around 3 watersheds, and you can also see the breakdown of reference versus removal sites within each of those watersheds. And I want to point out especially that this data includes sites from the Dolores River, Restoration Project, which was in partnership with the BLM and RiversEdge West. So for our data analysis, we constructed general linear mixed models that incorporated removal variables, biotic variables, and abiotic variables, so things like climate, water, roads, etc. And the way we constructed those models was using stepwise selection, starting with a lot of variables, and then after we had our significant variables, we conducted separate univariate analyses to show what was going on there. And we also removed 0 change rows for all of this. So if there was no salix prior or after removal, we did not include those. And to talk a little bit more about our removal variables, so we had site type, first of all, which included sort of the primary type of removal that was done at that site. And then we also incorporated undesirable and desirable references there. So that sort of included, each of these sites had one kind of primary type of restoration work that was done to remove tamaracks. And then we also just had our reference sites in that same variable category. And we also looked at whether any herbicide was used at any point, and also whether there was active plant introduction done, and that includes planting and seeding. It is important to note that we don't have data on what exactly was planted, so it wasn't necessarily salix, but we included it in the models. And then our dependent variables, so we did also look at final year cover. We're not going to be talking about that today. We're just going to be looking at cover change over time. And then, so we separately looked at combined cover of all Salix species, and then also pulled out just cover of Salix exigua. The reason for that was that even though the flycatcher can nest in any of these species of Salix, exigua is the dominant Salix species in these sites. It was present in 83% of them, and it's also functionally distinct from many other species. It can reproduce vegetatively, so we wanted to consider it on its own, and then also see if we, would see the same patterns or different patterns when we look at that one separately from all of the Salix species put together. So the results of our model selections, this is what the output kind of looked like. So we were interested in looking at adjusted R² of these to see like how well these models are predicting the variation in Salix cover at the end, or change in Salix cover rather. So we looked at marginal R² because that is what just is looking at the fixed effects, so the variables we were actually interested in. We did have random effects as well, but we weren't interested in those. And so you can see here that this particular starting conditions led to 5 different models. And so this was just looking at Salix exigua cover change. And so we had 5 different models, partially because our climate variables were intercorrelated, and so we had sort of these factorial designs where we tried to just put one in each to start with. So we ended up seeing that herbicide use was important in a lot of these, site type was important in one of them, and then soil, electrical conductivity, and Salix exigua cover prior to treatment were both significant in all of those models. And then again, climate was pretty much broadly important, and we had a different climate variable in each model because they were intercorrelated, but those came out pretty frequently. And in general, just to really briefly summarize what we found with the final year cover, site type was pretty important in most of those. But then when we look at the change over time models, it really didn't come out very often. This was actually the only time when it came out in any of our change over time models. And so that kind of shows us that, you can have an ending point that is going to be largely based on your starting point, but the change is not necessarily as affected by how good the site was to begin with. So just to talk about a few results that were interesting from this. So we found that soil properties were very important. So looking at Salix exigua on its own, soil electrical conductivity came out as a significantly negative relationship. Looking at all the Salix species together, percent sand was a significant positive relationship. Again, both of these are in the mixed model, not necessarily significant on their own. And so we saw that saltier soils are generally bad, sandier soils are better, and that makes sense with what we know about the niches that Tamarix and Salix can occupy. And it also shows us that even after removal of Tamarix, the salt will stick around, and that is going to have a negative impact. So it's going to make it harder for Salix to regenerate even after the Tamarix is gone. The next result we found was that herbicide use generally has a pretty positive impact on Salix cover change over time. And you can see there is a lot of variation. Part of that is because there was a significant interaction with climate. So generally, when there was more precipitation, you got kind of a better benefit from the herbicide use. If there wasn't enough precipitation, then it didn't matter as much. But yeah, both of these were significant. And again, just to reiterate, this was any herbicide used at any time. So also we found that existing Salix exigua cover has a negative relationship with change in Salix exigua cover. So if you already had a lot, it's not really going to be possible to get much more. So it seems like it exists in kind of a small niche that can be filled. And after a while, that intraspecific competition becomes more important than any kind of, increased reproductive potential that you would have had by already having some there. So when we looked at just removal methods in general to see if there were broad differences, there really weren't any individual primary methods of Tamarix removal that were associated with higher cover of overall Salix or Salix exigua. However, when we looked at just cut stump sites, we do see a proportional negative relationship between Tamarix and Salix cover change. So as you lose Tamarix, you are more likely to gain Salix. Unfortunately, that's also vice versa, and we see both. And there is a lot of variation in this. A lot of them are clustered around the 0 mark with not very large gains in Tamarix, but in some cases we can see that you can get a lot of Salix without really losing much Tamarix, and you can also lose Salix without changing anything about the Tamarix. So we saw this trend overall, but it's important to note that there's a lot of variation and it's not a clean relationship. And then when we look at overall with all of the removal method types, we don't see that same proportional relationship at all. It was not significant. And so when we look at planting methods, we didn't see a significant effect of planting or seeding. There was a lot of variation, and part of that, again, is probably just because we don't know exactly what was planted or seeded in each site. And in fact, if we had something other than Salix planted, then we would expect to see less Salix just due to competition. So to circle back to our questions, first of all, is it possible to increase Salix cover in such a way that we don't have a net loss? So we did see that proportional increase after cut stump removal, but it did not compensate for the loss in Tamarix cover. And also, when we just look in general, reducing Tamarix cover did not reliably increase Salix cover. So for that, we really can't say that we can, that there is a way to compensate for it perfectly. And when we look at specific restoration methods associated with increased Salix cover, we did not see a strong effect of any of those primary removal methods. Again, in cut stump sites, we saw that decrease that was associated with an increase in Salix. Again, that was driven in part by sites that got worse or, sites that just kind of had changes separately from that. And overall, herbicide use was beneficial. There was an interaction with climate. So then when we're talking about environmental variables, soil properties are strong predictors of cover change for both. Overall, we really see that Salix and Tamarix just occupy different niches. Tamarix is much more of a generalist. Salix has a much narrower niche. And also, these trajectories are still incremental and ongoing, even years after removal. And we haven't necessarily reached an equilibrium point with any of this. So just some final thoughts on all this. So in general, yes, Tamarix removal does result in a net loss of flycatcher habitat. And herbicide use is overall associated with more positive change. Again, we don't have a really good idea of how, what aspect of the herbicide use is most important, but overall it has a positive change. Removal methods on their own are not strong predictors of salix cover change in those cut stump sites, even though we did see, a proportional negative relationship. Tamarix was not fully replaced by salix. Soil properties were very important, but overall, really, we just can't expect salix to grow in areas where either it never grew or where conditions have since changed and it no longer can fill that niche. So some management implications of this. It is true that the biocontrol beetle does pose a danger to flycatcher habitat based on our findings. We can't count on Tamarix removal to promote salix cover. So if the goal is just salix regeneration and there is no flycatcher nesting in that area, then it makes sense to target Tamarix removal in cooler, wetter areas with sandier and less salty soils. And herbicide also generally seems to improve regeneration of salix. So with that, I'd like to acknowledge, first of all, that the data that we used for this project was funded by the Marie Curie Actions Research Fellowship Program, as well as the NSF Coupled Natural Human Systems Fellowship Program. And we also want to thank a few people for helping us out with development of our project and feedback along the way. So with that, thank you for watching, and we're happy to answer any questions.
And I'll be the one taking questions since, unfortunately, Alex Goez and Anna Scher were not able to attend today's meeting.
Thanks, Ian. Thanks for being with us.
No worries.
Looks like we have one question here. What is the water table factor in the change in salix following restoration? And then second part, was that measured at the sites?
Unfortunately, we did not. There was no hydrologic data incorporated into this data set. We didn't have any of that available. And that probably is, you could argue, you could argue that's probably the biggest weakness of the study, and probably incorporating hydrologic data in the future in future research would be really important.
Great. Next question, you mentioned site type as a variable in your data analysis. Can you elaborate on what types of sites were included?
What types of sites? So I think that site type meant it was like the specific removal methods involved, and whether it was like, what type of removal method was it, or whether it was kind of a reference site. And then there was two types of basically controls, which we had were like, high quality site control and then kind of degraded low quality site control. Right. And most of the difference you saw in the data was between reference and removal sites rather than between like different removal methods. So that really goes back to indicating that if you want to predict salix abundance, environmental factors are typically more important.
Okay. Next question, do you have any data on what percentage, a range of lost Tamarix habitat was replaced by salix?
What percentage was replaced in total? I don't believe we do. It was more as like for each site, how did the percent cover at each respond? I don't think we have like the total range data, but Professor Scher might have that. So if you're curious about that, that might be a good idea to email
her. I have a question. So in your conclusions, it said that, I may be paraphrasing it, but herbicide was a factor in the recovery of salix. Is this a direct correlation of using herbicide to control the Tamarix? And then I guess my second part of the question is did it account for all types of herbicide use on the Tamarix, like cut stump or foliar application or, but
I believe it counts for all types. I don't think it's like specific to just one method, but I think with the removal data, the fact that there was a significant relationship between change in Tamarack and Salix for cut stump. I mean, this is just probably me. This is just my kind of takeaway from it. I would probably conclude that cut stump removal would probably be the most effective.
Okay.
But you'd probably need more data to conclusively, go with that claim.
Next question, did the models incorporate any data on initial Tamarack density prior to the removal?
Yes, we did do that, and that wasn't as, I don't believe that was as big of a factor.
Okay.
But I'm not, I could be wrong about that. That'd probably be another good question for Professor Scher and Alex.
Ian, thanks so much for joining us for the questions section and helping present this information. We really appreciate you joining us today.
Yeah, no worries.
Okay, we're going to keep it moving along. I'd like to welcome Professor Cynthia Dott with Fort Lewis College. She has a presentation on a snapshot repeated riparian vegetation change in response to altered flow regimes and geomorphology in the San Juan River in southeast Utah.
Hi, welcome. We're here to tell you a story about a river with a snapshot
repeated. And this is about the San Juan River, which you can see was scoured to the bottom here. Well, okay, scoured in 1890s.
So we'll talk about riparian vegetation change in response to altered flow regimes and geomorphology on the San Juan River. I'm Cynthia Dott.
And I'm Gary Giannini, and we're at Fort Lewis College in Durango, Colorado.
We'd like to start by acknowledging that the land where we speak from today at Fort Lewis College and the region where we study this beautiful river is the ancestral land and territories of the Nuchu, Apache, Pueblos, Hopi, Zuni, and the Diné Nation.
We think it's important to provide this acknowledgement because the narratives of this land and region have long been told from one dominant perspective without full acknowledgement of the tribes who lived on this land.
Thank you for your attention and respect in acknowledging this important history. So our big question is, how did this on the left?
And what you can see there is the scoured channel of the San Juan River with small vegetation in 73, sorry. And then 47 years later, this year, turns into this jungle in the same stretch.
So here's an outline to that answer. We'll give you a little bit of the geographic setting and then talk about geomorphology and hydrology, and then use that as a way to explain the sequential waves of vegetation colonization we've seen along this river and end with some takeaway points. This is the study area in the red box near the town of Bluff, Utah, and you can see there's Fort Lewis College on the Animas River in Durango as part of the San Juan drainage. Our study area extends from just upstream of Bluff down to right above Lake Powell on the San Juan River.
And this is a Google Earth image. We want to highlight 3 types of canyon reaches for you. Alluvial reaches with broad floodplains, like you see here, shown in blue. Bedrock canyon reaches, highlighted in yellow here, as in this long reach here. And then those that are influenced by Lake Powell sediment out here in red.
The alluvial reaches look like this. They're wide floodplains, not really constrained by bedrock. And in this case, lots of room for the river channel to move back and forth, at least before vegetation, versus the constrained canyon reaches, which are about an order of magnitude smaller, narrower, and they're constrained by bedrock and often have bedrock channels and minimal sediment.
So when we think about the hydrologic setting of the San Juan River over the last 120 years or even a little bit longer, there are 2 main things that you want to notice. One is these large floods like 148,000 CFS in 1911. And note that as we go forward towards the future, those just stop. And the biggest thing we've had since 1980 is around 20,000 to 22,000. So that's a major thing. The other thing in the bottom of this slide is we have these alternations of wetter or drier than average periods. And important for plant establishment, as it turns out, was the Dust Bowl period here in the 1930s.
Okay. So to look at that hydrographic record a little more, here's the record of peak flows on the San Juan River. And you can see that pre-dam, the peak flows were about twice as large as they now are post-dam. It's pretty impressive. And then if we look at minimum flows, notice that minimum flows after the dam are about twice as large on average as they were before the dam. And during that Dust Bowl period, sometimes the river dried up. So I'd like to talk now about sequential waves of vegetation colonization in four sort of different time periods. The earliest time period is really setting the stage with scouring floods. Then we go to a period that was drier than average. Then we go to the post-dam period, which included a wetter than average period. And then we get to the present time where we find ourselves in the midst of a mega drought.
So here are some of those. Again, here's that same alluvial reach. And in the late 1890s and really into the early 1900s, these huge scouring floods that cleaned the alluvial reaches. And here's another picture of the alluvial reach right near Bluff, Utah. See this very, very wide, unvegetated floodplain, just little, probably willows right along the edge.
And then if we move down river a little ways, now we're looking upstream on the San Juan from near Butler Wash. And here we are in 1961, which is pre-dam still, but after most of that really dry period. You can see that there are cottonwood stands that are more dense along the shoreline. And on the islands and bars, we're starting to have colonization by willow and tamarisk. And the timing of tamarisk arrival in this area is a little uncertain, but probably sometime between the 30s and 40s. If we stay at this location now and jump forward in time to 2020, notice, it's pretty obvious, a huge change in vegetation. Now we're many years post-dam, and we're into this period of sustained low flows. We still have cottonwood stands. They're much larger now. Lots of Russian olive, that light greenish gray color, and some tamarisk is visible there. Now, if we move downstream a little ways further, we're at a popular hiking location, and here we are in 1961, so we're pre-dam again. And you can see in the background on that point that there is some tamarisk colonizing, probably some cottonwood and willow as well. But notice that the beaches are still fairly open. If we stay at this location, but now go to the river and look back towards that same shoreline, this is a photo from NAU's Klein Library donated by George Billingsley. There's George with baby Ryan and his wife Susan. In 1978, you can see that we're now post-dam. It's been a part of a wetter period. The cottonwood stand is fairly well established. There's tamarisk, zone of tamarisk. And most important in this image are the young Russian olive trees becoming established on the bank. If we stay at this location now, keep an eye on that knob, and go to 2020, you can see that the Russian olive is now hugely dense.
Okay, and if we also stay in the alluvial reaches and look here, just 3 years after the massive 1911 flood, you can see that this alluvial reach, it's just about to enter the canyon, but this alluvial reach is completely scoured with a few surviving cottonwoods. If we move forward in time to 1979, you can see there's a lot more vegetation, big cottonwood groves here, and then the sandbars are becoming stabilized and colonized by tamarisk and willow. Probably some small Russian olives out there, but we can't resolve those in this view. Then I went up on this on a Four Corners Geological Society field trip, and we took some pictures. And this shows the amazing encroachment of Russian olive in this side of the valley. You should note that if you go on this side, you need a permit from the Navajo Nation to hike.
Now let's move to the canyon reaches. And here's an 1894 photo showing that the scouring floods that were happening during this time period scoured the canyons too, and really left no vegetation, just these beautiful sandy beaches. If we stay in this location but move down to river level, here we are looking upstream from that same general location. And even in the 1950s, we're still pre-dam, but quite a bit of time has passed, there's no vegetation. If we now move to 2020, there's a lot of shrubby vegetation along the banks, mostly willow and tamarisk, some phragmites, and occasionally also Russian olive down in this part of the canyon. Tamarisk in this part of the canyon, based on tree ring counts established around 1968.
So in the bedrock sections of the canyon, this is pre-dam but post-drought, you can still see that the canyon is quite scoured, and there might be some small willows trying to establish here. But note where this man's head is. And if we come to this fall, Cynthia is standing where that fellow was, and it's a very, very different view. Phragmites right along the river, mostly coyote willow here, and a few dying tamarisk up high.
Based on ring counts, the oldest willows we see in this lower part of the canyon probably became established around 1997.
And then in the area that's influenced by Lake Powell sediment, which is actually built up, and river sediment has also built up in here, there are these amazing plant communities that are stabilizing the bars. And this is mostly willow and baccharis, and here a very, very large Russian olive. And what's amazing is these appear to be being chewed by porcupines.
Indeed. So to sum up those waves of vegetation colonization, we had frequent large floods in the early period, including that 1911 flood. And during that time, some cottonwoods survived, but towards the end of the flood regime, cottonwoods became established on the shoreline and expanded. Willows started colonizing the bars and islands as the large floods subsided. And sometime during this period, during drier conditions, tamarisk also began to colonize those bars and islands. After Navajo Dam, we see tamarisk establishing down in the narrow canyons. And Russian olive shows up, interestingly, pretty soon after Tropical Storm Norma caused a big flood on the San Juan in the 70s. Russian olive was probably there already, but that flood probably washed seeds in from upstream, and we saw a big increase in density. And then lastly, the most and an interesting new development is that we have more willow establishment down in the narrow canyon reaches, we think as a result of lower flows and no big floods to scour them away. Tamarisk is dying back in that portion of the canyon, and we're now seeing more willow and even Russian olive now showing up down in the canyon.
And probably part of that willow establishment or encroachment now is because of the higher low flows during the summer months.
Right. So part of the answer to how we got from this to this
is this decrease in scouring floods, which we showed you data on. Part of that's because there's Navajo Dam upstream. Part of that is nature doesn't appear to be dishing up the huge storms that we had at the beginning of the last century.
And the drier than average conditions that we've had a few different times during this period have permitted initial vegetation growth and bank armoring. And then the higher minimum flows kind of further the growth of that vegetation on the banks.
So the net result of these combined factors, both geomorphic and hydrologic and vegetative, is that native and exotic woody vegetation has greatly increased along the San Juan River corridor.
So some takeaway points. Dense riparian vegetation is likely here to stay. Even if we did get a large scouring flood, it would take a lot to remove that vegetation. So how we plan and manage in this system is going to determine whether it's dominated by native species, like cottonwoods, or whether it will be dominated by non-native Russian olive, which we've seen expanding exponentially in the time we've been spending on the San Juan River. So with that, I think we will pause for questions.
And thanks so much for listening to our talk. And we'll go to questions, and we'd welcome your thoughts and questions. So this is us doing field work on the San Juan, and that's one of our field assistants down here.
Thanks so much for your attention.
All right. Thank you so much, Cynthia. Gary, thank you so much. Sorry I did not get a chance to introduce you, but greatly appreciate your contributions and your presentation. So just to comment, the photos that you have in the presentation looks like they're a big hit. So wow, that's quite a contrast, seeing the old San Juan River. I'll just kick it off with a question here, and I know this is part of the story in other rivers. So you have a big wide river, narrowing, increase in natives and non-natives. Is there a significant impact on the San Juan River on aquatic organisms, fisheries, because of this, right, where we're getting less complexity in these systems? Is that the story of the San Juan as well?
Yeah, as far as I know, the channel complexity is really decreased, like you said, Rusty. So native fish in the river, native fish populations are struggling for a lot of reasons, as they are in other regional rivers. I don't know too much about the details of that, but definitely impacts.
Okay, looks like we have some questions. Did you investigate major watershed sediment changes, i.e. channelization upstream, heavy grazing, etc.?
Not in detail, but certainly there are changes in sediment loading. Interesting, a lot of the sediment input on that portion of the San Juan River comes in below Navajo Dam, so there's probably less reduction in sediment on the San Juan River than on some places like the Colorado below Glen Canyon Dam, for example. I don't know, do you want to add anything?
No, I think that's right.
Yeah.
Okay. Any thoughts on how this new dense riparian vegetation has affected bird populations, good or bad, especially for those species that need this vegetation?
Yeah, cool. That's a great question. I remember running into an ornithologist on the river a few years ago, and he said, we have so much better bird habitat along the river now than we used to. Kind of interesting, a little ironic perhaps. So I don't have specific data, but I think the one, I don't know about Swiffle, if there are southwest flycatchers in that region. I know that yellow-billed cuckoo habitat was considered possible in Glen Canyon National Monument, so the lower portion of the river, but I don't believe any have actually been seen there. I think they require more tall canopy trees like cottonwoods than exist in that part of the canyon.
Okay. Do you have any thoughts on management actions to pursue?
It's a big project, but I think if we don't want that system dominated by Russian olive, it's going to require active removal. And there will always be Russian olive in there, I'm sure, but
Right.
if we don't actively remove it, it's moving down canyon pretty rapidly. It used to only be in the alluvial reaches, and now you see big stands down in the canyons, and as we showed, even all the way down onto the lake sediment. So it is moving downstream, and it's going to require some pretty active intervention.
Yeah, and I just add that the efforts to reduce Russian olive upstream all the way down from Navajo Dam are really going to be critical, because it's a continual seed source for the reaches we're studying. So really it takes an integrated watershed approach. You need somebody to really want to make rocking chairs out of Russian olive or
Thank you. Did the beetle have any effect on tamarisk, and did Russian olive fill the void where tamarisk has died back?
Yeah, interesting. People ask me that a lot. The beetles have had a big impact on tamarisk on the San Juan River. We see a lot of tamarisk actual dieback, like dead trees, as well as just defoliation. I don't think loss of tamarisk has allowed an increase in Russian olive. I think the Russian olive was coming on anyway. So Russian olive is increasing, tamarisk is decreasing, but I don't think that that's causal. I don't think, because Russian olive comes up in the understory, it's a shade tolerant species, and so it's coming up under the willows in a lot of places actually. But the tamarisk decline is a real thing, and that's presumably mostly because of beetle.
Okay. Cynthia and Gary, sorry we can't get to all the questions. I'm going to keep it moving along. There are a few left about flows out of Navajo, Russian olive removal, maintaining side channels. Maybe if you guys will be around with us today, we can set up a breakout room where you can answer further questions. Cynthia and Gary,
Yeah.
thank you so much.
Yeah, thanks. Thanks, Rusty. Be glad to do that. And
Okay.
I'll look at the questions on the chat as well.
Okay, great. Thank you.
Thank you.
Okay, next up, I'd like to welcome Lisa Markovchik. I think I got that right, Lisa. I want to welcome Lisa. I want to welcome Lisa from Northern Arizona University. She's going to be speaking on riparian habitat and wildlife. What's mycorrhizae got to do with it? So thank you, Lisa, for being here, and we'll have her presentation.
Hi, everyone. My name is Lisa Markovchik, and I am a PhD candidate at NAU in Flagstaff. And I am here to talk to you today about habitat and wildlife, and what does mycorrhizal fungi have to do with that? So you might wonder why, when you guys probably care a lot about these guys, I want to talk to you about these guys. And I'm going to use the southwestern willow flycatcher as an example today. So we know that swiftwood fecundity is declining due to nest heating. And we know that tamarisk and or mixed vegetation is hotter and drier than native vegetation. And also that we're staring down the barrel of a lot more warm and dry weather due to climate change. And interestingly, mycorrhizal fungi actually contribute to water and evaporative services in the local area that they're found. So on the top left here, you can see an illustration just of the stomatal opening in a plant leaf. And we're just trying to illustrate the fact that mycorrhizal fungi actually help plants moderate their water use efficiency through a few different means, but particularly through modulating that stomatal opening and closing. And then on the bottom right, you can see in the little call-out box number 2 there are examples of mycorrhizal fungi transferring water. So on the left, transferring it within the mycorrhizal network. And then on the right-hand side of that circle, leaking it out into the upper soil layers, as is pictured below. And what you see below that is just a hyphal tip, where the water has been stained with a fluorescent stain. So you can actually see the hyphal tip in fluorescent green, as well as a little drop of water coming out of it. So before we go too far, let's just talk about what mycorrhizae are. They're an ancient fungal symbiosis with nearly all land plants. They actually helped land plants move to land. And there's two main types that are relevant in our riparian communities, particularly for cottonwoods and willows, because they use actually both types, but also for any of the other trees in the riparian community. And in many of our habitats, they'll usually use either one or the other. So AMF is our buscular mycorrhizal fungi, pictured on the bottom left there inside a plant root cell. And then EMF, or ectomycorrhizal fungi, which occur on the outside of the roots. And you've seen some examples of those already, and there's one right there. These guys all receive photosynthate in exchange for a variety of resources or services that they provide to the plants. And that list of services includes things like boosting survival, growth, resiliency in the face of stressors. Again, the water services that we just talked about, so providing water access and helping improve water use efficiency for the plant, providing access to nutrients and improved plant health, also providing a variety of techniques of pest control, and helping to reduce the toxics that are absorbed from the soil to the plants. I could go on, but let's stop there. The important piece is that these impacts on all of these services from mycorrhizal fungi are not negligible. They are very frequently 50% or more impact on, for instance, rates of survival or growth and so on. However, as we've begun to get more familiar with the kingdom fungi, which is still relatively underexplored compared to other kingdoms, we have learned that land use changes and different management actions affect mycorrhizal communities too, both in diversity as well as abundance. So for instance, invasive vegetation, and I'm going to talk particularly about tamarisk, but also things like habitat fragmentation, land use change, fuel management activities, herbicide application, many of the things that we do every day and often have other types of beneficial responses and that's the reason we're doing them, can impact our mycorrhizal communities. So you may have seen some of the results from my field trials. I'm just going to talk about it briefly. We worked out on the Little Colorado River doing tamarisk removal and cottonwood and willow restoration. And we set it up in this way. So we had either local cottonwoods from a nearby population or the site itself. And then we had what we called transfer cottonwoods, which were from a slightly warmer area, slightly further away from the site that we were restoring. We had areas that either had a tamarisk history and soil legacy or did not within the site. And then we either added a sort of live soil inoculum of mycorrhizal fungi from a site that hadn't had tamarisk and was more intact. And we either added that or didn't add that to those treatments in a fully factorial design. And what we found was pretty striking. So tamarisk legacy definitely reduced cottonwood survival. And the mycorrhizal inoculation improved survival, but the treatments and the tree source interacted. So I wondered, what values and services are we losing for plants, habitats, and other dependent species when mycorrhizal fungi in the community decrease? Can we restore those values as well by restoring the mycorrhizal community? So during the pandemic, we have been busy with a greenhouse trial, and it was basically set up in the exact same way, using the same tree genotypes and the same soil, but just in the greenhouse where we could control a variety of factors a little better, such as soil moisture and things like that, and make sure they were consistent. Borrowed a gas exchange system, the Leghorst 6800, so that we could take some gas exchange measurements and look at the ecophysiology of the plants. And what we found was that for local trees, tamarisk legacy reduced the water use efficiency. So on the left-hand side of the graph, you can see the treatments where we did not have tamarisk soil. So sort of the status quo, if you will, both inoculated or not. And on the right-hand side of every graph, you're going to see the plants that were planted in the tamarisk legacy soil. Those dark bars are always going to represent mycorrhizal inoculation. The light bars are without mycorrhizal inoculation. And what you see here is that in Tamarisk legacy soil, that light bar of water use efficiency went down. And then when we added mycorrhizal fungi, that water use efficiency went back up. Survival showed a very similar pattern. Why might that be? So Tamarisk legacy seemed to actually cause the trees to produce beyond their means. So if we look at a graph of the photosynthesis, we can see that on the right-hand side of the graph, that photosynthesis in the Tamarisk soil shoots way up without mycorrhizae additions, and then sort of goes back to the normal level when we add that mycorrhizal inoculation. For the transfer trees, the results were actually almost exactly the opposite. So again, on the left-hand side of the graph, sort of the status quo water use efficiency. On the right-hand side of the graph, we can see what happens in the Tamarisk legacy soil without mycorrhizal inoculation. That water use efficiency shoots way up. When we add mycorrhizae, it brings it back to that status quo level again. And we can see that the survival is also tracking that pattern. This seems to be because Tamarisk legacy is actually shutting down the plant production without mycorrhizal inoculation. So you can see here in the Tamarisk legacy soil, that photosynthesis, the light bar on the right-hand side goes way down. When we add mycorrhizal inoculation, it bumps back up to sort of that status quo level again. So in summary, due to the Tamarisk legacy and reduced mycorrhizal communities, local trees overproduced, reducing their water use efficiency and having negative impacts on their survival. Transfer trees did the opposite. They actually shut down in that Tamarisk soil, which improved their water use efficiency and survival, but would essentially shut down their photosynthesis and resulting growth. In both cases, the mycorrhizae actually stabilized the situation. So they stabilized both the photosynthesis and the water use efficiency while maintaining survival at a better rate. So why would trees react oppositely to this mycorrhizal loss and or restoration? What we've been learning from the literature is that the pairings between the trees, the soil, and the fungi really matter. And so in this case, transfers were better paired with the mycorrhizae, and locals were better matched with the soil. Also, the plant growth strategies probably differed because the ones from warmer sites would have had a slightly different growth strategy. Appropriate mycorrhizal inoculation basically really matters. And when I say appropriate, what I'm talking about is this pairing between plants, soil, and mycorrhizae. So we can actually get neutral to negative effects with poor pairings. And we just really need to think about those hard and long when we do our mycorrhizal inoculations and restorations. So I wanted to know if Tamarisk reduced mycorrhizal communities cause plants to shut down production or die, then shouldn't mycorrhizae boost Swiffle habitat suitability, and would we be able to see that in a habitat suitability model? And so James Tracy, who's presented his Swiffle habitat suitability model at this conference in previous years, it's a GIS-based site-specific model, went ahead and entertained my questions and added a restoration project of cottonwoods and willows to an area in the model that was previously Tamarisk. And what we did was we went ahead and added the survival parameters and growth parameters from the literature for cottonwoods and willows. And then we also added on the bottom here, you can see some of the references for what mycorrhizal inoculation did for plants, whether it was biomass or survival and those kinds of things, to the model. So we incorporated that into the model, and we had a treatment without mycorrhizal inoculation and with. And you can see on the left-hand side here that with mycorrhizal inoculation, after 6 years, we actually have 1.2 hectares of suitable habitat at this site for Swiffle, and that would be corresponding to that purple line on the graph where you can see the canopy cover increased much more quickly than the blue line, which is if we did not have mycorrhizal inoculation. And in fact, after 6 years post-restoration, if we did not include mycorrhizal fungi, we still had 0 hectares of suitable swiftwell habitat. So invasive reduced mycorrhizal communities also reduce plant survival and growth, and as a result are going to be reducing suitable habitat for other species. And we're just talking here right now. We haven't added anything but growth and survival metrics to the model. So we're purely talking about growth and survival. We're not even yet including things like what effects that has on shading, what effects that has on canopy cooling with the evaporative services being provided. Also, appropriate mycorrhizal restoration is stabilizing plant survival and growth and increasing suitable habitat for other species. Again, just purely based on mycorrhizal impacts to plant survival and growth, not even yet incorporating shading, canopy cooling, evaporative services, and so on. And so just like these guys who all have very specific needs and requirements and perform different roles within the ecosystem, we need to remember that these mycorrhizal communities, all the different species and taxa in there, have similarly very different needs, roles, and provide different services. So as part of that, we're actually going to be using stable isotopes next with tracking water movement through mycorrhizal hyphae in an attempt to go ahead and identify which mycorrhizae seem to be accessing water and providing it to plants, and also to see whether the tamarisk legacy is actually reducing not just the mycorrhizal community and abundance, but actually negatively impacting the mycorrhizae that might be providing these water services. I just want to say a huge thank you to everyone for attending today. It's been a pleasure talking, well, to or with you. And this research would simply not be possible without an army of folks. I can't even get the list on the slide anymore. So I'm just very thankful to all of them as well. And also a note that if you're interested in incorporating mycorrhizal restoration into a project, I would love to hear from you. I'm trying to get us set up so that we can provide that service more regularly to folks. And I would love to chat with you about it. And again, thank you so much. And hopefully we have some time for questions.
All right, great. Thank you, Lisa. Really appreciate the information. I want to remind everybody that Lisa will be joining us and having a workshop on mycorrhizal fungi and restoration in the southwestern U.S. Tomorrow on Thursday. So they'll have a whole workshop that afternoon. So I want to remind everybody to join Lisa and the gang for further information. We do have some questions, Lisa. We'll just go ahead and get to it. What's the mechanism of the legacy effect? Is it just a lack of the bank of fungal spores, or has tamarisk done something to the soils that inhibits mycorrhizal fungi?
Oh, great question. Thanks, Rusty, and the folks yielding the questions. Yeah, so we've actually been doing some research on that. What it looks like is that there are probably multiple mechanisms. For instance, one could be the salinity that folks were just talking about in relation to a soil legacy from tamarisk. Another, though, that we know of is that a lot of invasives, not just tamarisk, but tamarisk is one example of it, that they actually don't use mycorrhizal fungi. So one of the sort of things that helps them move around the globe and take over is that they either don't have a relationship with mycorrhizal fungi that they're dependent on, or they can kind of release themselves from it. So they might actually have mycorrhizal fungi in their home native locations, but when they travel the globe, they usually don't, or often don't bring any mycorrhizal fungi with them and don't need to, and they're perfectly happy to operate without it. So what happens as a result is that once you have an invasive species in the soil for a certain amount of time and it's kind of taken over, you have this legacy in the soil where the mycorrhizal fungi have essentially been starved out because they really haven't been able to proliferate and reproduce and multiply in that soil for an extended period of time.
Okay, sorry, I'm going to back up one. Yasmin had a question I looked over. What is the average difference in mycorrhizal fungi levels between Tamarisk and native dominated communities?
That's a great question that I don't know off the top of my head. And I'm just gonna look here really quick. Yasmin, I'd be happy to get back to you as well. But we do have that in our workshop slides. And so that's what I'm trying to look for here. Yeah. Let's see. Sorry, we can go ahead and take another question while I look for that slide.
Okay.
If you'd like.
How do you determine the appropriate mycorrhizal pairing? And what does the on-ground application look like? I've used mycorrhizal coating on seeds, and can it be broadcast over a site? A multi-part question.
Yeah, so gosh, there's a lot packed into that question, and we're going to cover it in a lot more detail on Thursday,
Okay.
where we'll have 2.5 hours to talk about it. But there are a whole bunch of factors that we're sort of finding are important from some of the field experiments and some of the literature. So all sorts of things like tree genotype and where the trees came from and what sort of community they've been co-adapted with at that site, as well as things like soil characteristics that those fungi themselves would then be adapted to. So there's probably a lot of factors to talk about, more than we would have time for here, but you can, you can actually do restoration of the mycorrhizal community, which is what we'll be talking about on Thursday. I haven't tried broadcasting it as a coating, although I'd love to chat with folks who have and compare notes. Usually what we do is the most sort of inexpensive and simplest form, which is to gather that mycorrhizal community from a place that we know hasn't had as much of a history of disturbance and herbicide use and things like that, and tamarisk legacy or other invasives. And then we'll actually bulk it up in the greenhouse with things that we know mycorrhizae like. And then we'll go ahead and use that. And I'm just going to go back to the other question here. So for instance, it's probably going to vary from invasive to invasive species, what the effects are. But in a particular study by Meinhardt and Gehring in 2012, for the arbuscular mycorrhizal fungi, colonization went from, say, 55% to about 30% when there was a tamarisk legacy or neighbor. And for ectomycorrhizal fungi, colonization went from 60% to 30%. So it actually halved in both cases.
All right. Well, Lisa, I'm really sorry we got to move on. There's another number of other questions about mycorrhizae. I would welcome everybody to the workshop on the 25th from 2 to 4:30 p.m. Hopefully we can get to some more of these questions. Lisa, we're happy to set up a breakout room if we need immediate questions and if people can't join the workshop. So let us know if we can facilitate that. Lisa, thanks so much.
Hey, Rusty, just before you go, so I'm unfortunately stuck in lab teaching for today and tomorrow.
Okay.
But I'd be happy to welcome more folks to the workshop. And also I'll put my email in the chat box because I'd love to hear from you if you are interested in incorporating mycorrhizae and that kind of thing. Thanks so much.
Very good, Lisa. Thanks so much. Okay, we're going to move on to our last presentation of the morning session. I'd like to welcome Wally McFarland from Utah State University. Wally's going to talk about a riverscape network assessment of Lahontan cutthroat trout habitat. So Wally, thanks for joining us, and we'll go ahead and get his presentation and do some questions afterwards.
Right, good afternoon. I'm Wally McFarland, and over the next 15 minutes, I'll provide an overview of our Riverscapes network assessment of Lahontan cutthroat trout habitat. This is a 2-year project, which is initiated by the U.S. Fish and Wildlife Service. So there's a core recovery team that contracted out with us to do an assessment of habitat. I would like to acknowledge my co-authors, Nick Bowis of Utah State University, and also of Ecological Research, and Philip Bailey of North Arrow Research. So this is an outline of what I'll cover in this presentation. So first, I'd like to help provide a conceptual background for our habitat assessment approach, and I'll provide an overview of a fairly new stream evolution model that describes different stages of a stream that a stream might go through over time, ranging from incised and disconnected from its floodplain to a stage with multiple threaded channels and high lateral connectivity, which is referred to as stage 0. Next, I'll make a case for why lateral connectivity is an important indicator of fish habitat quality. Next, I'll describe how lateral connectivity will be assessed in this project using the riparian condition assessment tool. And I'll finish off the presentation by providing the primary applications of this habitat assessment. So I don't know how many of you have seen these stream evolution models. This is one by Kluwer and Thorne in 2014. And you'll notice as you go through around this, these describe different stages from intact to degraded, and then going back to widening and improving, and back to a multi-threaded stage, which we refer to as anastomosing or stage 0, where there's high connectivity with the valley bottom and high resilience and habitat diversity. In this incised state, channels and floodplains are disconnected, and these stages have limited fish habitat quality, complexity. Here's an example of incised and highly degraded stream. I'm sure many of you in your travels throughout the Intermountain West, the Southwest, have seen streams like this. They're heavily incised, they're down cut. The floodplain is now converted to upland species. There's very limited riparian vegetation. Here's an example looking more from an aerial view of this incised, disconnected floodplain stage. The idea here is this has been degraded by down cutting, by incision. And so the historic valley bottom was all active floodplain. Now the current condition is you have this incised channel with a very narrow active floodplain with a wide inactive floodplain. So this anastomosing state at the top, there are examples throughout the Intermountain West of this. And in this case, this is the Marys River in Nevada, part of the Lahan Cutthroat Habitat Assessment Area. And just looking at this here, we have a valley bottom out to here. This whole area is active floodplain. We have multiple threaded channels. We have high lateral connectivity, and we have complex trout habitat. So this is what we're shooting for. This is our target. So next, how are we going to go about how and why are we using lateral connectivity as a proxy for fish? Kind of to build our case, I have two examples. The first is Bridge Creek. This project involved studying how can beaver dams and beaver dam analogs accelerate the process of incision recovery, and can this benefit fish populations? So that idea that currently varying sized stream, and through these restoration activities where 240 beaver dams were built along with BDAs. And then the fish response, the steelhead response is pretty remarkable. 168% increase in abundance, 52% increase in survival, and 172% increase in productivity. Another example that's cutthroat fish. This is in Birch Creek, Idaho. In this project, we reintroduced 9 beaver, and over the course of 3 years, they built 140 dams and just transformed the riverscape. So this is the first time in 40 years there's been perennial flow throughout the system. Prior to that, it would dry up in the fall or late summer, fall. So trout have increased 50-fold from previous surveys, and beaver complexes have increased 10-fold. So pretty remarkable transformation of this riverscape, and at the same time, the effects on trout populations. So how do we plan to go about assessing lateral connectivity and using it as a proxy for habitat quality, fish habitat quality? So pretty simple. The idea is that the current extent of riparian vegetation with the valley bottom will be used as a proxy for lateral connectivity. So in this example, this is high lateral connectivity. So this would be scored very high. So the higher the proportion riparian vegetation, the higher the lateral connectivity. And we'll use the riparian condition assessment toolbox to do this assessment. The riparian condition toolbox, maybe some of you have heard of it, RCAT. It's comprised of first a valley bottom extraction tool. The idea here is the valley bottom is this area from this hill slope to this hill slope. It includes the floodplain and the channels. Riparian vegetation departure, RVD, is the second tool, which we'll talk about, and then the riparian vegetation conversion type, as well as the riparian condition assessment. These are currently being run with readily available nationwide data, and we're fortunate enough next year to be able to run these same models with NDVI-derived products from Ken McGuire at Desert Research Institute. And he's developed a riparian vegetation map for the state of Nevada, which is a very useful product. So let's go into a little bit of more detail on each of these tools. So on the valley bottom extraction tool, the idea here is that there's thresholds that we can use within a drainage area, which will have different slopes. So for example, in these lower valley bottoms, you might have quite a bit flatter slopes than compared to, say, these tributaries here, these main tributaries, compared to these headwaters. So the idea here is that we can derive valley bottoms throughout the whole drainage area if we use these thresholds and these different slope estimates. So the valley bottom becomes input into the riparian vegetation conversion, riparian vegetation departure. So this is simply O to E, so observed versus expected. So in this case, we have a valley bottom. We have it broken up into these polygons, these segments. And so here's our existing vegetation divided by historic, so our O to E, and we come up with a riparian vegetation departure. And these values are showing how much they've changed from historic. So in this case, large departures are 66% or greater. And this has been run throughout the state of Utah. So the riparian vegetation conversion type, RVCT, just looks at that historic riparian and provides a output of what it's transformed to. In this case, there's conversion to agriculture throughout this example area. In the places you folks work, likely there'll be a lot of conversion to invasive. So it gives you that idea of what changes we're seeing from historic to current. And then the riparian condition assessment, RCA, brings in RVD, so riparian vegetation departure, and land use intensity. This might be agriculture versus pasture versus just open area. And then importantly, we also look at floodplain connectivity. And here we look at how roads, railroads, and in some cases levees and dikes have broken up, fragmented the valley bottom. So in this case, red represents disconnected floodplains and blue are still intact. And these are all combined within a fuzzy inference system to provide a output that's reach-based, but can be run throughout a whole riverscape. So in this case, what we see throughout this study area is we have some, still intact areas in good condition versus those that are in poor condition. In this case, probably driven by agriculture. And then in the headwaters, because headwaters really don't have a valley bottom, we just stick with confined, impacted, or unimpacted. So I'm just going to provide some quick examples of the riparian condition assessment tool in Utah. And this case study is another Birch Creek. This is in southern Utah. And this is, first we can see the valley bottoms are in green. We have a zoom in area, which is a restoration area. This is a Bonneville cutthroat trout habitat restoration project and sage grouse habitat. So you can see out here, there's been a lot of pinyon juniper removal. Within the valley bottom, there's been a lot of vegetation encroachment and incision. So if we look at the tool outputs, the riparian vegetation departure shows large departures in the upper reach, and then some changes from minor departure to major to minor to major. And this corresponds with the vegetation conversion type. We see here that we have conifer encroachment significant up here, and then less here, and then in the lower, there's just really not much change since historic. And then if we look at the riparian condition assessment, we see poor condition up here. This is this corresponds with the highly incised area where it's disconnected from its floodplain. And then we have moderate to good down in this area. I got to turn off my phone really fast. So that's the riparian condition assessment. The primary applications of this riverscape assessment and the projects you guys will work on will probably have similar applications, but we see it as helping to identify both conservation and restoration areas, and those areas that should be prioritized because they're likely going to have the greatest benefit for, in this case, the Lahontan cutthroat trout. And this helps prioritize where restoration work is done and then ongoing monitoring. I think this is a case for the importance of assessments. A lot of us talk about, restoration and monitoring, but a first step is to assess what the condition is currently. So it's very important. And this helps with the establishment of recovery potential. As we look at what the current state is, we can also gauge where we can get gains, where we can have the greatest recovery potential. And then finally, this is very important information in developing conservation and restoration plans. So for in this case, Juan cutthroat trout, this could be applied to other species. And the idea is these can be really important inputs for those plans and the assessments that follow. With that, I'd like to acknowledge my co-authors and the development team and those that have helped throughout the development of RCAT. Many people to thank. And with that, I'll leave it open for questions. Thank you.
All right, thanks, Wally. Appreciate you being here and appreciate you presenting on this issue. We'll take a couple questions and then break for lunch. First one is, how will the NDVI product be incorporated?
Yeah, that's a great question. How that product will be incorporated is we'll use that as the existing vegetation layer. So what Desert Research Institute has done a really nice job of is harvesting a lot of the Landsat data from as far back as Landsat goes in the late 70s. And so we'll be able to use that for existing or different snapshots in time to look at how the riparian areas and the vegetation within them have changed.
Great, thank you. Wally, what are the applications for some of these tools that you presented? Are people able to access these and use them for different watersheds, or will this be scaled to different watersheds? How will that work in the future?
Yeah, right now we're working on actually getting funding to do a west-wide run of the tool. So from the 100th meridian west. And so if that does occur, we'll have widespread coverage. But to date, we have information, as I mentioned in the presentation, for Utah. We've run it in various watersheds in Idaho, Oregon, and Washington, but it's a little a mixed coverage there. So hopefully as funding comes in, this will be something that will be available widespread.
Okay. Is there any work to incorporate potential impacts from erosion or site-specific conditions into the model? We worked with RCAT before and tried to incorporate some of that information into our analysis.
Yeah, the flexibility of the tool is dependent on what data you have, and so the tool it can be modified if there's additional inputs. Right now, one thing we're working on is bringing in more information if there's existing levees or elevated roads or information like that, as far as to improve the floodplain connectivity end of things. But I imagine you could also incorporate information on erosion or incision or factors like that as well.
Okay. Well, Wally, I think that's going to take us up to the lunch break. Thanks so much for joining us and presenting those tools.
Yeah, thanks for the opportunity. Thank you. Great session, by the way. Really appreciate all the talks were awesome.
Thank you, and thank you to all the presenters and the great questions. So that takes us to lunch break. So we will have, I believe, a breakout room with Professor Dot to answer any of those questions. So you guys can click on your breakout rooms, I believe, at the bottom of your toolbar to access some of those. So we will join back here in just shy of an hour. So thanks everybody. We'll see you in a bit.